So SAR-bisPSMA is not only a diagnostic, it's also a therapeutic. You currently have your secure trial ongoing, and that's currently in the cohort expansion phase. Maybe if you can provide an update and some background as to the design of that trial, the data that we might expect to see in the next sort of 6 and 12 months, and then sort of plans for the future.
Yeah, this SAR-bisPSMA is so unique in the market, and it's unique in many different ways. But the innovation behind it first,
Mhm.
making a new molecule, but having the opportunity to use the same molecule for imaging and therapy is fantastic. Just shifting the isotope, the perfect pairing, as we call it. Uh, but it's not the only part that we enjoy doing science. We love science,
Mhm.
and and for some reason, some of the market doesn't like science. This is in biotech.
Yep.
So we want to make sure our science is outstanding in order to win.
Mhm.
Um, and so when we went about doing a therapy trial, we wanted to make sure something critical that had been missed by the entire market, including, and this, when we talk about the market, it's Pluvicto.
Yep.
And that's the number one player. That's the one we have to beat at this point in time. But we'll discuss another player which is of interest to us. That data came out recently. But in the Pluvicto setting, we want to be a Pluvicto buster.
Mhm.
And we've done the hard work to date to be that, because we've built a monomer, and it wasn't any good.
Mhm.
And then we built a bit superior product. That was the goal. Now, the other part we wanted to do was a dose escalation and dose optimization.
Mhm.
And we went through that process. 4 gigabecquerels, 8 gigabecquerels, 12 gigabecquerels, single dose. And we started to see even phenomenal data there,
Mhm.
even at the 4 gigabecquerel dose. The 8 gigabecquerels was amazing. And that's where we saw the first patient we ever dosed. What it was going to be a dose that we thought was going to be at least therapeutic was the 8 gigabecquerels. But to have a complete response in that first patient we ever treated at that level, it was an incredible result.
Mhm.
That patient lives today without cancer.
Mhm.
And this is a few years back.
Mm.
So, and then we're up in the 12 gigabecquerels, and we multi-dosed at the 12 gigabecquerels. And still, it didn't matter how hard we pushed, even at that 12 gigabecquerels. We see the Telix safety data come out recently, where there's 50% of the patients with heme tox above grade 3.
Mm.
We weren't even close. We had one patient with grade 4.
Mhm.
That's it. So, so the safety was there, but we started to push some safety parameters that we're not comfortable with. Group like Telix is comfortable with this, great. We would can the project if it was like that. And so our safety data was much better, but we wanted to make sure that it was incredibly safe,
Mhm.
because we want to beat Pluvicto, and the efficacy stacked up. And that's when we pulled it back at 8 gigs. And that 8 gigs now is being multi-dosed. And we've got a slightly different dosing regime,
Mhm.
because even in the conversations you have with Louise Emmett and Oliver Sartor, the adaptive dosing schedule is far better.
Mhm.
Dosing when you need it is far better than just dosing up front.
Yep.
Okay? So, and I think the entire community is comfortable with that view. The issue that you have, if you have that, you can't run a trial, because you don't know when you're going to dose. And then suddenly you have these, yes, OS is pushed out for years and years and years. And how do you actually make that work?
Yep.
So we've got something in between. It's between 6 weeks and then the sort of 12 weeks leading into an additional week. So 13 weeks, 6 to
Mhm.
13 weeks, you're dosing within that period of time, subject to how heme tox is going, how some other measures are going, to make it absolutely safe to use is really the goal.
Mhm.
It's never going to be 100%, and we're even seeing some grade 1s, but we see the grade 1s reverse in the clinical trial. So that was our second goal: do some great science on the structure of the protocol, and then we've got a great molecule, great isotope, great structure, so that now the questions that we're asking, and the key one is, what is our phase 3 dosing going to look like?
Mm.
Now we're generating that data.
Mhm.
We work a little bit different to some organizations. We release our data as we go.
Mhm.
We had the safety review committee at the 8 gigs. Fantastic result today. We said there was another response where undetectable PSA
Mhm.
and undetectable PSMA in imaging. Fantastic. Our share price, I think, fell 20% on that because people don't seem to value miracles anymore. A patient has that sort of response
Mm.
and their AEs are in the grade 1 and get better is a miracle.
Mhm.
I'm a scientist saying that, so I don't believe in miracles. I believe in hard work, dedication, and focus and attention to the patients. And that's the outcome we had. We just released then the 5th patient just recently, same outcome.
Mhm.
No PSA, and PSA is a very important factor
Mhm.
as the first part. If there's no PSA, and all patients know this because they have a prostatectomy or definitive therapy, their PSA drops. The cancer is removed, it drops, right? Hopefully to 0. The patient group then it comes back because the cells are there, but then they're nascent for a period of time and they come out. And then we want to treat biochemical recurrence, but then when it gets worse, we want to treat with these therapies. To have that response in patients, you saw the amount of previous therapies that they had failed,
Mhm.
some of which are blockbuster therapies, and we have that response in a patient with grade 1 AEs and they get better. It's a miracle. It's fantastic. Radiopharm is heading into that space. We want to make sure we have the best agent. We see Pluvicto as the best. We want to be a Pluvicto buster. That is our goal, and our data is stacking up. Even on the small number of doses, we're not doing 6 doses 6 weeks apart at this point in time because we're having these responses after 2, 3, potentially 4 doses.
Mhm.
Incredible response. But we also are watching out for competing groups, competing products in the innovation phase.
Mhm.
We saw the product release some data recently, was the Bayer group, the Trillium asset.
Yep.
Now, we like the group that invented that product, John Babbage and his group. They are very similar to us. They're the hard work at the benchtop, smart people. We've known each other for a long time, and they have an alpha asset, which the AEs are a little worse than ours, but the efficacy was looking good. And so when we're looking at ourselves in the market, we want to go as early as possible as a beta. And we want the concept, and it's something that we've discussed internally a lot and also with our key opinion leaders. We want to hit like an alpha,
Yep.
but have the safety profile of a beta.
Yep.
This is something the alphas can't necessarily do.
Yep.
And certainly from the beta perspective, all we have is Pluvicto that made up a dose and ran with it. So our plan is to complete the expanded cohort. We'll get all of that data in. Recruitment is going well in that. We wanted to make sure that we measured the first patients at 8 gigabecquerels multidosing in case we have to change it. We have now confidence that that looks like the dose that we're going to use in a phase 3. We're going to run that out now, that trial, looking to close recruitment this year on that, and we'll look at the data. Then we'll make a call on what the phase 3 looks like and press forward with the phase 3. If our data is as outstanding as it is so far, then we're expecting the phase 3 to be relatively more on the smaller side rather than the larger side because the difference can be seen in a smaller group of patients, and we'll keep our eye on the competition. As I said, we don't see a lot of competition, even though people say, I'm in PSMA, I'm this and that. Show us the data. Show us the efficacy data, then we know where we're at. And the only one that we can see that's in development at the moment was a recent one. But let's play. We think we've done the hard work there. Once again, when you're preparing for something, you've done it for so long, you build this level of confidence in your product. We did the hard work on the dosing. Now it's time to complete that trial and show the data.
Given this is the Science Hat podcast, I wanted to ask a couple of questions that I'm intrigued by. Thinking about the asset, the molecule, but the molecule coupled with the 67 copper, and you want to be a Pluvicto buster, what element do you think is most important to it being a Pluvicto buster?
Great question. The mechanism of action for radiopharmaceuticals is area under the curve. So the most important part is the amount of product you get in the lesion and how long it stays there. That was our original goal. We wanted to build a product because the PSMA agents have moderate uptake and then washed out. And we think it was because of the recycling of that receptor and how that binding occurs.
Mhm.
Our goal was to overcome that. So in the basic work, we saw we're able to achieve that outcome 2 to 3 times at the same imaging time point. But then it increased even over time. Now, the mechanism we found was a very large amount of the product internalizing.
Mhm.
So when we look at the grassroots, what we're looking at is to actually have a more enhanced mechanism of action, more product in the lesion, and it stays there over time. So utilizing copper-67, it's a great isotope. We think the betas, as we said, was are safer in that context. Um, and that's purely over the punch.
Mhm.
Alphas hit hard.
Yep.
And we know there's offsite targeting with these agents. We want to hit softer on the tissues which are there to stay
Mhm.
and hard on the tissues that we want to go. So that massive amount of internalization in the lesions where there's a large amount of expression of the target is a great position to be in. We seem to be having that, and that's why we're having more of the grade 1 responses on the AEs, but having an absolute killing effect on the lesions.
Yep.
That's what we see as the Pluvicto buster.
Yep.
Pluvicto hasn't necessarily done that level of work. There's a level of internalization work and dosimetry and the like, but we think that's the mechanism that we can overcome. Data will tell the story.
Yep.
We'll let the data shine. Let's finish the expanded cohort. 8 gigabecquerels, as we said, is a nice, safe amount. Multi-dosing at that with a slightly different dosing schedule. I think we've done the hard work. But you never know until you get into the grand final how you're going to perform. I think we've done the hard work at the moment to go up against these types of agents. And as I mentioned, we know where the competition, do we think antibodies are the way? No, they're not even close on the safety profile, and let alone nobody's showing efficacy.
Mhm.
Once we see this other data read out, then we'll know where we sit.
Mhm.
The data's looking really good so far. Remembering that we use the same agent as the diagnostic, and we know how good the diagnostic is.
Mm.
So that's just feeding the same agent into a therapy protocol. Yes, it's slightly different because we're dosing at much higher levels and, there's a few differences in that mix, but same molecule, same targeting.
And just finally on the therapy, there is a lot of competition in prostate cancer. Outside of, the radioligand therapies, there's also sort of the T-cell engagers. There's different, broader bispecifics and trispecifics. And patient recruitment is always the question. So you said secure, it's recruiting nicely. And, just thinking about in the future for a phase 3, you still feel confident in being able to recruit?
Yeah, it's a good question. Like when you develop new products, you can't constantly live in fear of someone curing cancer. We have a need to produce better products. And so when the Janux data come in, for instance, I was getting messaged by an insto saying, "Oh, look, this Alan, what's going on here?" I said, "It's early.
Mm.
Let's see how the data reads out." And then,
Mhm.
that came out.
Yep.
It was a little bit more in line with what our expectations were. So we can only do what we do.
Mhm.
We've done the hard work, as I said. We don't see anything at this point in time we're fearing to any great degree. I mentioned what we're keeping our eye on. There are other products, other mechanisms of action, which is fantastic for prostate cancer patients.
Yep.
And because you have another mechanism of action, it means you can potentially then still use our product in that patient group. But we would love to see better advances.
Mm.
I'm a scientist. I'm trying to make the world a better place. We want others to be part of that. If we were to monopolize there and just say, no, only use our product, or our product was so toxic, for instance, and then, patients can't use other products because of how toxic it is. And we're seeing, as I said, we're seeing that in some other products. And even some of the things we've seen in radiopharm, the J&J strategy, I think was terrible, to be frank. The outcome of that, yes, the hard work was done, the antibody was done, but an antibody with an alpha and the result that we had was terrible for the patients. We can do better. And I think the small molecule approach is working really well. What Pluvicto was there. We think Pluvicto is an okay product. It's good. It's a good first step. But can we do better? Yes. Are there other areas that are going to advance? Yes. And hopefully then in the overall development paradigm, we can reduce the side effects, which, considering that, first line therapy is blocking a man's testosterone, hopefully we can do that better with better products. And then increase the efficacy so that they can have the responses that we've seen in our patients, recently. Undetectable PSA, undetectable PSMA imaging, and adverse events in the grade 1 and get better, and have patients live for 3, 4, 5, 6, however many years without cancer, is a fantastic outcome.